Thank you for your interest in eDNA monitoring of feral swine. My name is Anna Mangan. I'm with the National Wildlife Research Center in Fort Collins, Colorado. And today I'll be talking about a case study in southern Indiana. I'd like to start by acknowledging my co-authors, Amberly Hauger and Lee Humberg from Indiana Wildlife Services, and Tim Smyser and Tony Piaggio from the National Wildlife Research Center in Fort Collins, Colorado. So for this audience, I know you're all familiar with feral swine, and you probably know that in the US, we've got a feral swine problem. As you can see from the shaded counties in this distribution map from 2019, they are ubiquitous, and they've been found in as many as 38 states. In some areas of the country, they have really dense populations. The worst parts of Texas, for example, have an estimated 8 to 16 feral swine per square mile, which is actually more than the population density of people in Alaska, Montana, or Wyoming. But in other parts of the country, it's a different story. States like New Mexico, Colorado, and Indiana have feral swine that are few and far between. And in these areas, wildlife managers are working hard to keep feral swine from gaining a foothold on new invasion fronts or to eradicate small populations. And part of the challenge for these managers is knowing when they've removed every last individual from the landscape. And this is so important, partially because of the incredible reproductive capacity of feral swine. So they reach sexual maturity at an early age. They have litters of 4 to 8 piglets and usually reproduce or have multiple litters each year. So having just a few pigs remaining on the landscape can put managers back to square one in no time at all. And this is also a problem because of how damaging feral swine can be. Invasive wild pigs can damage agriculture, timber, and property. And they also spread diseases like brucellosis, influenza A, or pseudorabies to humans, livestock, and other animals. And from an ecological perspective, they destroy habitat, negatively alter water sources, and compete directly and indirectly with native species for resources. So how do we find these last few pigs on the landscape when the consequences of missing them are so high? Well, especially in these post-control situations, environmental DNA can be an effective tool where we've got low densities. We can literally be chasing the last few pigs around a vast landscape. And current collection methods can be relatively easy to use. Field biologists can collect a few water samples while they're out conducting their other field duties. And eDNA can also be useful for remote or hard to reach areas that are time consuming or costly to monitor with other methods. So for those of you not familiar with eDNA or environmental DNA, let me back up just a little bit here. In this process, we're collecting fragments or complete DNA cells that have been shed by our target organism and are suspended in the water. So eDNA has been used for threatened species like hellbenders or aquatic invasive species like Asian carp, but our lab was the first to develop an eDNA assay for a terrestrial mammal. And in previous studies, our lab demonstrated an effective sampling method for collecting water samples and a laboratory assay that can reliably detect feral swine DNA from water bodies. So since that time, we've implemented this tool into field operations. For example, a successful project has been going on in eradication of areas in New Mexico for about 3 years. And especially in the drier months, we know that feral swine are drawn to limited or ephemeral water sources, like the one we see circled here. They'll use these water sources to drink or wallow, and in the process, shed DNA, which we can then collect. And based on our eDNA assay, we can find detections or non-detections to identify hot spots on the landscape. And managers in New Mexico have been using these hotspots as a way to help prioritize where to put effort and resources. And they've successfully removed wild pigs from areas that we identified with eDNA. So these methods have been successful in arid ecosystems, but as you know, feral swine are distributed across a variety of ecosystems in the US. So we wanted to know, can we use this non-invasive monitoring tool in other novel environments, or is it really best suited for arid climates? So to answer this question, we designed a small pilot study in southern Indiana, where our primary objective was to determine if our eDNA sampling and laboratory assay strategy were efficacious in this novel environment. So Indiana has had a small population of feral swine for roughly about 30 years. And in 2014, the National Feral Swine Damage Management Program started the process of eradication in this state. And in the last 5 years, we've been particularly successful at removing pigs from this area. We've been utilizing a combination of surveillance methods like cameras, tracks, and bait stations, as well as aerial observations. And now we're getting to the same problem that we've seen in other states, where it's difficult to find these last few animals on a vast landscape. But unlike our example in New Mexico, where we have limited water resources, southern Indiana is not water limited. This area actually sees a lot of rainfall in spring and fall, and flooding is not uncommon. So for this pilot study, we selected 4 locations where feral swine presence was unknown. Those are these black boxes on the map shown here. And then 8 locations where feral swine are known to inhabit, these green circles. And these are in Lawrence and Jackson counties. So you may notice that the known sites are clustered around 4 geographical areas. And since we wanted to be sure that we had the chance to capture eDNA from these known pig sites, we decided to sample these areas twice. But due to field constraints, we weren't able to sample the exact location twice. So we've called each sampling occasion a separate site. So when samples were collected, we also documented any pig sign that was recently around and estimated the approximate age of that sign. Samples were collected from mid-October to early December in 2019, and we collected 10 water samples from each site by collecting 45 mils of water and then adding 15 mils of a preservation buffer to a Nalgene bottle. We also collected a negative control at each site to monitor field contamination. We also implemented an adaptive laboratory analysis to maximize our probability of detection, yet to minimize the lab resources that this study required. So we started with 3 samples per site at a time, and we extracted each sample 2 times using a Dionysi Maricon food kit, and then ran 6 ddPCR replicates. We reported all results as detection, non-detection, or presence-absence of the feral swine DNA. And again, for this iterative process, we looked at the results as we went through each set of 3 samples per site. So if we got to a point where we saw 3 or more positive replicates, we would stop and classify that site as positive. If we had less than 3 positive replicates, we would go back and run the next 3 samples from a site through that extraction and ddPCR process. And this would continue until we either had 3 or more detections, positive detections, or we went through all of the samples. So in total, we collected 120 water samples and a negative control from each site. But based on all of our extraction and ddPCR replicates, we ended up with more than 1,300 replicates and negative controls. So you can see that even for a small pilot study, we really ramped up the lab effort quickly. So in the next few slides, I'll take you through our results table, and we'll start with the samples from known pig activity sites. So you can see the site name here, along with the approximate age of the pig sign at the time of collection. And we further divided that into a less than 2 weeks or more than 2 week age of the sign. And we arrived at this threshold because in our previous research with captive pigs, we found that DNA could be detected from a single pig. It would persist in the water for about a 2-week period, which is this gray slope that you're seeing here. Interestingly, in this same study, we also found that if multiple pigs were using a water source, the DNA would persist in the water a lot longer. So that's this black bar along the top. We actually found that DNA could be detected in 100% of our samples all the way out to the end of our 2-week study. And our models estimated that the DNA would persist for approximately 72 days when multiple pigs were using the water. So back to our results table, we now see the proportion of positive replicates. And this is simply just the number of replicates that amplified divided the number of total replicates that were run on ddPCR for that site. And we use this proportion to classify the site as either positive or not detected. The first site shown here actually showed signs of field contamination, or we had amplification in the field negative control. And this is exactly why we collect those samples. We don't want to be misidentifying or misinterpreting results as false positives. So we discontinued any further analysis from that site. But of the remaining sites, we had 5 positives and only 2 sites where we didn't detect feral swine DNA, even after we ran all of the samples from those locations. Interestingly, these 2 sites were in the greater than 2-week sign classification here. And these 2 Ratcliffe sites had pretty old wallows when we collected samples. So it's possible that the pigs had just visited and too much time had passed before we sampled to actually detect any DNA. It's also possible that the pigs we know are in this area just never actually used the actual water sources that we sampled. So we just missed the opportunity to capture DNA. Okay, now let's look at the unknown sites. Interestingly, here we actually found 2 positive sites. And this is exciting because we hadn't had any record of pig activity, either tracks or cameras or reports, in about 6 to 10 months from either of these sites. So after finding these eDNA positives, we've actually ramped up scouting and began placing more trail cameras and doing increased aerial surveys in hopes that we can find these last few animals. So when we look at the full table of results, it's important to note that not all positive sites are the same. As you can see, we have a wide variation in the proportion of positive replicates. So some sites like these top 2 are really hot, where we have feral swine DNA that was detected in lots of replicates and really quickly, which means that we had to run fewer samples to get those positive sites. The Bear and Bear 2 sites, for example, we only ran that first set of 3 samples per each site to get these positive classifications. On the other hand, the Nolan and Nolan 2 sites had far lower proportion of positive replicates. And we ended up having to run all 10 samples, analyze all 10 samples, and we just barely met our threshold for having 3 or more positive replicates to classify those sites as positive. So importantly, most of our sites showed agreement between our initial knowledge of pig presence in the field and our eDNA results. I've added these red pluses to the map here to show the eDNA positive sites. Furthermore, we found these 2 areas where we need to be boosting monitoring efforts, where we previously thought that feral swine were absent. So as we consider the value of eDNA monitoring, we can see that it gives us insight into where feral swine are on the landscape and helps us prioritize where we might be best spending resources and effort to remove these small pockets of pigs. So while we're really excited to see the success of this pilot study, we have to remind ourselves that no monitoring tool is perfect. Even things like cameras and tracking have limitations. And eDNA is really just another tool that can facilitate detection when we have low densities. But it doesn't work in all situations. And we identified some challenges during this pilot study. So first, it's clear that we need to monitor contamination continuously. We always report these contamination issues back to field collaborators so we can identify if equipment is contaminated or if perhaps more training may be needed on collection protocols. We also found that eDNA persistence can be kind of tricky. Again, there were 2 sites where we failed to detect feral swine, even in our known sites. And again, this may be because too much time had passed since the last pig visit and the DNA had degraded, or it could be due to other site characteristics. But on the other hand, we don't want to be collecting samples that may contain eDNA from pigs that were recently removed. So again, if we think about our previous research that showed DNA can persist for about 2 weeks, In areas of active pig removal, we use this rule of thumb of about a 1 month grace period before we do any resampling. And finally, even with our adaptive laboratory analysis, it became clear that significant laboratory effort was required for all of this processing. And currently, as the sole eDNA analyst, this can mean that there are um, slow turnaround times to look at results. But the success of this pilot study really is encouraging. It gives us another tool in the feral swine management toolbox, and we're planning to use eDNA along with other surveillance methods in Indiana in the future. We also found that this adaptive laboratory analysis approach does a great job at saving resources. If we had continued running all the samples from every single site, we would have had at least 400 more replicates to analyze. And finally, and perhaps most importantly, the study gives our program confidence to expand eDNA beyond arid ecosystems and into other important areas of the country. So, for example, we're currently considering eDNA projects in Missouri, where eDNA would be helpful for confirming if elimination zones are indeed feral swine free. So we're always moving eDNA forward, and we'll be using the lessons that we learned here as we pursue new areas and ecosystems, which may be good candidates for this surveillance method. So we'd like to thank the National Feral Swine Damage Management Program for their support of eDNA research, and Indiana Wildlife Services, especially Maddie Pfaff and Travis Buckle, for their assistance in site selection and sample collection. And finally, I'd like to thank the National Wildlife Research Center Wildlife Genetics team for their support and initial development of these methods. If you'd like to leave any comments in the video, you can ask questions there, or you can contact me directly for questions. Thank you very much.